our work in this field called translational bioinformatics. So I'm a faculty member at the Baker Computational Health Sciences Institute at UCSF. I've been at UCSF for about 5.5 years, but I guess I'll spend the first few minutes just telling you guys about me and my career path and how I got to where I am today. So I was born in Moscow, Russia, and I moved to the Bay Area in 1995, which is a while ago. I attended a public high school, Abraham Lincoln High School in San Francisco, which I think recently has been renamed. And then I did my undergraduate and graduate work at Stanford in bioinformatics. And I really got interested in this intersection between biology and computer science at the end of high school. I was taking this amazing class. I don't know, I might have, no, I don't have a slide on that. In biotech, it was called a biotechnology class, and we got a chance to do very cool experiments like make bacteria glow in the dark by gene transfection and do PCR, and I thought it was awesome. I thought genetics was really cool, but I realized I wasn't very good in the lab. I did know, however, that I was pretty good in math, so I wanted to bridge my interest in math and my interest in science. So I asked my teacher, and he said, "There's this field of bioinformatics, and when when I'll get to college, I will figure it out." So that's exactly what I did. After graduate school, I actually spent a few years working at Pfizer, a pharmaceutical company, and we're going to be talking about drug discovery today, so that's very, very relevant. Prior to returning to the academic world and becoming a professor at UCSF. So I always like to start by giving a little bit of background on, genetics and genomics. So do any of you guys know who these two guys are? Just unmute yourself. Anybody? There might be some, oh yes, okay. Watson and Crick from Ryan, yes, that's correct. So these are the guys who came up, discovered DNA, but also came up with the central dogma. This is James Watson and Francis Crick. And the whole idea is, we have DNA that's in chromosomes in our cells, and it goes through the process of replication. Then it gets transcribed to RNA and then translated to protein. And all living things are made out of proteins. And we study all of these molecules. We study DNA, we study RNA, we study protein. And I think as part of this presentation, you'll see that everything is important, and it's really looking at, different types of molecular measurements that allow us to get a better sense of what might be going on in disease. And specifically, we'll be focusing a lot on RNA. DNA, as you guys know, is stored in chromosomes. We have 22 autosomes and then X and Y chromosomes. Well, some of us X and X. You can really think of, once you look at the basis of DNA, you can think of it as a really long string of letters. So, and we're lucky enough to have the technology to be able to measure those and read those out. Where I started in terms of research is this field of comparative genomics. And this is just a figure of a phylogenetic tree where there, of course, as you can see, different species, and two species are closer to each other if they're related to each other. So for instance, we are very closely related to chimps, macaques, baboons, and other monkeys. And then, we have some relationship to rats and mice and rabbits, and then cats and dogs, and then fish, much further away. But the whole idea is that you can use the sequence, and this is showing the proteomic sequence, to be able to figure out which regions of the genome haven't changed much. So here you can see the regions that the sequence that hasn't changed too much is highlighted in the same color. And the idea is, if these regions of the genome have not changed very much throughout evolution, then maybe they are functional or they're important in some way. Because if they were mutated and those mutations would be detrimental, then they would be lost throughout the evolutionary tree. So these are the types of questions that I started working on, and this was a while ago. And really, it is the sequencing of the human genome in 2001 that has led to many of these, to us being able to answer many of these questions. But that's on the genetic side, that's on the DNA, right? And the protein. But what about that in between molecule, the RNA that I was mentioning before? So there is this idea, this type of data, molecular data called gene expression. And the idea behind this measurement is that it's a global snapshot of what is going on in a cell at a given time. So while the genome sequence tells you what might be going on, the expression tells you what actually is going on in a cell. So which genes are turned on and which genes are turned off. We have, people argue about this all the time, but we have about 20 to 30,000 genes, which translate into over 1 million proteins. And we can measure a few thousand proteins reliably, but really we cannot measure 1 million proteins. However, we can measure the expression of those 25,000 genes or so pretty robustly. And there are different methods for doing that. There is a technology that has been around for quite a while called the microarray. And these are little glass chips where thousands of probes or little sequences are hybridized to that glass surface. And it takes advantage of the fact that both DNA and RNA, they're complementary. So if you have a probe and there's a complementary probe that is present in a given sample, then they're going to bind. And the more of that probe that's present, the more of it is going to bind to that hybridized attached probe, which will result in a signal once the chip is actually scanned. So the process for this is RNA is first extracted from a certain sample. It goes through fluorescent labeling, then it goes through hybridization and wash, then they're scanned and goes through signal processing, and then we get to the analysis part, which is what I'm mostly involved with. Another reason why I'm excited to be a computational biologist today is that there's tons of data in the public domain. And these are just examples of these resources that are available to anybody. So any one of you who is a little bit computer savvy can go online and start looking at these data sets, start downloading these data sets and playing around with them. So the Cancer Genome Atlas is an example. This is a collection of over 1,000 cancer samples that have been profiled with a variety of different technologies, including gene expression. So specifically, they have RNA-seq data. So that's a different way to get at the same type of data as I just described. They have proteomics data, which means the proteins. They have methylation, which is a modification of the genome. So many, many different molecular measurements. The 1,000 Genomes is a project to sequence individuals from different ethnic backgrounds. And it's a fantastic reference data set. GEO is the Gene Expression Omnibus, and we were talking about gene expression just now, but this database contains pretty much every single expression data set that has ever been generated. And because many of our studies are funded by the government, by the National Institutes of Health, we have to make, as scientists, we have to make our data publicly available. And that is a tremendous opportunity for anybody who has some computational ability to analyze data statistically. And it's fantastic. So in addition, people are coming up with creative ways to use these technologies because they're getting cheaper and cheaper. So the genome we just talked about, people have been able to sequence whole genomes since 2001. The transcriptome, that's the gene expression side of things. But also people can measure the antibodies. So looking at the immune system, looking at epigenetics, so that's the genetic modifications, looking at proteomics, and the microbiome, which is different bacteria that might be present in a certain sample. And it is really the intersection of all of these technologies that allows us to ask new questions and integrate this new data to study disease. And that's what my team at UCSF is doing. As we know that the cost of the, genome sequencing has gone down tremendously in the last 20 years, so has the cost of computation. I mean, these things that we all have are much, much more powerful than computers were only 5 or 10 years ago. So it's an amazing time to be a computational biologist. So in my group, we're interested in figuring out how to use these different types of data to identify better diagnostics markers, to understand disease a little bit better, and to impact therapeutics. And within the therapeutic realm, we're really interested in finding new uses for drugs that are already FDA approved. And that's the whole idea of drug repurposing. But before I get into that, I want to pause and see if you guys have any questions about anything I've mentioned before. Don't be shy. Okay. Either I lost all of you or everything is clear. Okay. What software is used to analyze the data? That's a great question. It depends. Great question, complicated answer. It depends. So I myself started out my career programming in C, which is a very low level language, and C++. But a lot of the work that we do now is in the statistical language R, which is a very high level language, has a lot of packages implemented and a lot of bioinformatics tools already implemented. And a lot of the work is done in Python as well. So if you're running more complex machine learning algorithms, then Python is the way to go. If you're doing more statistical analysis and data visualization, well, Python is pretty good for data visualization too, but R is better or maybe more preferred. So it really depends on what is the data and what is the question. Very little of what we do now is in the lower level languages like C++. So that maybe some of the early like genome processing steps like quality control and alignment are implemented in those languages, but Java with Java knowledge would be helpful. That's a good question. I actually myself have not programmed in Java very much. Maybe for visualization, but I would say Python and R are the two big ones. Excellent. Other questions? Okay. Well, I'll keep going, but I'll give you guys opportunities to ask more questions as we go along. So it takes a long time and it's very expensive to take a drug and bring it to market. And 90% of these drugs, they fail in early development. One solution to that is drug repurposing, which lowers the cost of development because these drugs are already FDA approved for something. So a lot of the safety testing has been carried out already. And it also, as a result, reduces the risk of failure. Do any of you guys know an example of a drug that has been repurposed? So with the idea that originally it was intended for a certain indication or a certain disease, but then used for something else. All right. Nobody. No examples. Thalidomide. That's one, yes. Any others? Okay, well, one of the most famous ones is Viagra, actually. I don't know if you guys know, originally it was developed for a cardiac indication, and then the nurses observed side effects, and then it was repurposed. So we want to do the same thing, but we want to do it more systematically than just by accident. And of course, now there's many more examples with COVID-19. So the methods that we're using here are, the method or the approach is called computational drug repurposing. And there are different ways to reach this goal. There are target-based drug repurposing using genetics. There is disease-based drug repurposing. So the idea behind the genetics piece is that if there are certain diseases that share genetic targets, then maybe a drug from one disease can be repurposed for another. The disease-based drug repurposing, the idea is maybe if two diseases manifest themselves similarly, then a drug for one disease from disease A could be used for disease B. And that has been, a good example of that is autoimmune diseases, because the immune system attacks something in the body for some reason, we don't know why. And people in many ways try drugs from, let's say, rheumatoid arthritis in another autoimmune disease, like ankylosing spondylitis or something like that. There are molecular modeling approaches which take advantage of the structure of a drug and the target and how they interact with each other. I will be honest, I'm not an expert in that at all, but I know there are many groups working on that a lot. And then there are finally network approaches and global signature approaches. And these really mean that one drug, one target, one disease is not sufficient for complex diseases. So that's what we wanted to explore. That's the approach that we ended up taking. And we wanted to ask, can we use public gene expression data? So we talked about gene expression quite a bit, to systematically predict relationships between drugs and diseases. So on the drug side, let's say you have certain cells that are treated with a compound and you have expression before and after. And same thing with the disease data. Let's say you have healthy individuals and disease individuals. So we can then elucidate which genes are involved in the disease and which genes are affected by the drug. And you can start to connect these drugs and diseases using gene expression as a proxy. And here, what we want to end up with is a matrix of diseases and drugs, where each cell of that matrix is a score of how likely a given drug is to treat a given disease. So as a computational scientist, what do I do first? I go out and look for data. So on the disease side, we leverage one of the databases I mentioned before, called the Gene Expression Omnibus, which contains over 2 million microarrays. Like I talked about those glass chips, over 2 million samples that were profiled with expression measurements. But the more impressive number is this 84,000. That's 84,000 individual experiments. That's more than any university or pharma laboratory could ever generate on their own. But as a computational scientist, you have access to all of that. And many of these are associated with disease. On the drug side, we used a data set called the connectivity map. And this was generated by the Broad Institute. And what they've done is they've taken a number of drugs. The first set was 164, and the second was a larger set of about 1,000 compounds. They put them on cancer cell lines, and they measured which genes are turned on and which genes are turned off after the treatment with the drug. So starting with the disease data, there's a lot of work that went into curating this data. And these are just some relevant references. First, identifying the disease experiments, then identifying which of those disease experiments have a healthy versus disease comparison. So in total, we ended up profiling about 100 diseases. So for each of those data sets, we have the disease individuals and the healthy controls, and we look at which genes are different between the two groups, statistically different. We do the same thing for the drug data. We have untreated samples and treated samples with a certain drug, and we look at which genes are perturbed by those drugs. And our hypothesis was very simple. If the disease gene expression signature is reversed by a drug, then maybe that drug could be used as a therapeutic. We saw situations where the signatures were similar and of course, when things were all over the place. So we developed a computational pipeline to take a disease expression signature and query it against the drug data, coming up with these drug disease scores, where the idea is that score would be positive if the drug is similar to the disease, and it would be negative if the drug is opposite to the disease. And really, we're interested in these negative scores because those are the ones where the drug is likely to reverse the expression signature of the disease. This is that matrix of drugs by diseases that I was mentioning before, where the drugs are on the x-axis and the diseases are on the y-axis. And really, we're interested in these negative scores, or the green ones, the ones where the drug profile is the opposite of the disease. But obviously, you see a lot of black here, which means no significant relationship. And some of the scores are positive, which means the drug has a profile that's similar to the disease. So this was very interesting, and we looked at various clusterings of drugs and diseases and saw things that made sense to us. But we really wanted to take a single disease and see if we can predict a therapeutic for it. So I will pause here for a second. Oh, I see there's a question. Great. How do you deal with outliers in the drug-disease relationships? So can you explain a little bit what you mean? Do you mean the outliers on the gene level when we look at the correlations? You can unmute yourself. Yeah, okay. So that will be taken into account in the calculation itself. Because if, let's say, you're looking for a strong correlation and you see that certain, so the simplest thing, if you think about plotting all the genes, well, we were doing this in the rank space. So if we plot all the ranks of the genes in the drug experiment versus the disease experiment, if the correlation is perfect, well, perfectly negative, then it will be on the reverse diagonal. And if they're outliers, then they will fall outside the diagonal and affect that correlation value, right? So then if things are all over the place, then just the relationship will not be significant, statistically significant. Does that make sense? So the outlier question in terms of the genes, that doesn't pose a problem. It will be taken into account as we do the computation itself. Okay, other questions on the method? Okay, no questions. Okay, I'll move on, but please stop me if you have any questions. Okay, so the single first application of this approach that we decided to focus on was Crohn's disease. So Crohn's disease is an inflammatory disease of the intestine that has an autoimmune component. It affects 500,000 people in North America, and there's no known pharmaceutical cure. The current solutions are to either reduce inflammation with anti-inflammatory drugs and corticosteroids, which have pretty bad side effects, and there are also surgical solutions that are pretty invasive. So looking at our therapeutic predictions for Crohn's, we saw that prednisolone, which is currently used in the clinic at least to alleviate symptoms of Crohn's, was one of the top hits. However, there was one other drug, topiramate, that had a better therapeutic score. So turns out topiramate is an anti-seizure drug. It suppresses the rapid and excessive firing of neurons at the beginning of a seizure, and it does so by enhancing GABA activation. It's used to treat epilepsy, bipolar disorder. It's an antidepressant, and it has been investigated as a potential treatment for obesity and type 2 diabetes. So looking at the expression signature for Crohn's here on the right-hand side, and then the expression signature for topiramate, the signal is not perfect. But if you squint, you can see that the genes that are upregulated in the disease, shown in green, are downregulated in the drug, and genes that are downregulated in the disease are upregulated in the drug. And when we look at the pathways, they make a lot of sense. So there's gastrointestinal disease, a lot of immune pathways, NF-kappa B, IL-15, B cells, inflammatory response, IL-10. So all of this makes biological sense. The signal is not perfect, but it's one of the best ones we've got. So we went out and we looked for an animal model for Crohn's disease and identified TNBS as such a model. It's fairly widely used both for IBD and Crohn's. And this is what a normal rat colon looks like. This is what a colon of an animal where the disease is induced look like. So you can see that a lot of the structure is just gone. So we started with a small validation study, and this was in collaboration with Jay Patricia at Stanford, that consisted of 18 animals. So healthy controls where the disease model is induced that were untreated, and disease model induced that were treated. We used a commonly tested dose for these animals and then collected the colon tissue to be scored by a pathologist. And this is what those preliminary results look like. So the score for the macroscopic damage score, which is the pathologist evaluated data, ranged from 0 to 6, with 0 being no inflammation, 1 and 2 some swelling and redness, 3 and 4 several ulcers, and 5 and 6 mild and severe necrosis. So here in green, we're showing the healthy animals. In red, where the disease model is induced, and in blue, where the disease, where they were treated with topiramate. So we saw a stark reduction in the inflammation in these animals, which was incredibly exciting to us and also quite hopeful. So these are just some images of the colon tissue. You can see there's a lot more inflammation in the untreated animals than animals that were treated with the drug. So then we did 2 other follow-up experiments with 2 independent groups. Here, in addition to all the previous groups, we also include prednisolone, which is the a current treatment. And we collected a lot of data. So clinical signs, pathology scores, histology, and endoscopy images. And this is what those data look like. So this is looking at clinical signs, more specifically, percent of animals with diarrhea. The greens are healthy animals who are just fine. And in red are animals where the disease model is induced. And in blue and purple is treatment with topiramate and prednisolone, respectively. So you can see that the inflammation and sort of the clinical signs go down with both treatments. However, when we look at the histology, which is the underlying inflammation in the tissue itself, we saw a significant reduction with topiramate, but we don't see a significant reduction with prednisolone treatment. And that goes for the microscopic as well as the gross pathology score. And these are representative histology and endoscopy images also. So then we wanted to go back and see whether the genes from our original signature contribute to this. So we tested a handful of genes, and this is an example of one that goes down in the disease model and is brought back up by the treatment. And this gene, IFI30, is upregulated in the disease model and then brought back down by the treatment. Again, not ideal, but at least it gave us some confidence in these results. So that was the first application of this method. Since then, we've applied it to a number of different conditions. I wanted to highlight one specific project that uses a slightly different idea for drug repurposing. And this was a project carried out by Anushree Sengupta, who was a high school intern in my lab. And now she's an undergraduate at Stanford. So she wanted to see whether we can use shared genetics between diseases to predict new uses for drugs. And she carried out all the computational analysis relating to this project. So here, she also wanted to link diseases and drugs, but instead of using the gene expression as we were doing before, she was using gene association. So that's the DNA measurement that I was talking about earlier. So she was able to connect the genes, the drugs which are shown in red with diseases that are shown in blue using the genes which are shown here in yellow. And these are just showing the drug and the disease nodes, and the size of that node is how many genes are associated with that feature. So she identified a ranked list of 618 drug disease pairs, and within top 100 of them, 89% of them were known indications. So whatever she was finding was real. But we wanted to take one example and see what it looks like in the real world. So the case study that she focused on was aminocarboic acid for peripheral artery disease, or PAD. So this drug is usually used for excessive post-surgical bleeding. And the disease is the narrowing of the peripheral arteries, which is a pretty chronic and painful condition. Turns out that this drug works by targeting a protein called LPA, and specifically prevents the formation of this lipoprotein, which has been linked to PAD. So then we wanted to go to the electronic medical records or the clinical data at UCSF, and we identified 83 patients who were diagnosed with PAD who were on this drug. And we looked at their blood pressure, and we saw that in the individuals on the drug, which are the cases, those are the guys in red or this reddish pink color, both the diastolic and systolic blood pressure was significantly lower. So again, this is not proving anything 100%, but it gives you an idea how you can use different lines of evidence to get to the same answer. And this clinical analysis was done by a postdoc on my team, Nadav Rapaport, who's now a professor in Israel. So as I mentioned, with this approach, this is the reference to the original paper, we've applied it to many different diseases. So dermatomyositis, cancer, various cancers actually, pregnancy outcomes like preterm birth, and our latest application is applying this approach to identify new compounds for COVID-19. But I'd like to pause here for a few minutes before we move on to this last piece and see if you guys have any questions. This data could be used by clinicians to determine the best treatment and avoid worsening the disease. Well, hopefully, right? I mean, I look at this type of analysis as a way to generate hypotheses that then they still need to be tested. So we've done some testing preclinically. There probably still needs to be at least a short clinical trial looking at the efficacy of these approaches, but it's a way to generate hypotheses quickly using the data that we have on hand now. What challenges did you face? Yes, Ryan, that's correct. The idea that it guides research, but also hopefully clinical practice moving forward. What challenges did you face during your study and how did you overcome them? Well, a big challenge in a lot of our studies is cleaning data, actually. Because we work with data that's publicly available, it's free, which is great, but it's often noisy. It's noisy because sometimes things are not as well annotated. It can be noisy because people make mistakes. There's a human factor here, right? So there's a lot of time that goes into cleaning data and understanding data. And I would say that's one of the biggest challenges. I mean, the other big one is figuring out what question to ask. And here, I mean, we were fortunate enough to come up with a question that was very interesting. Yeah. Okay, does this type of population, the genes, does the type of, yes, absolutely. Well, in expression, not as much as in genetics, but nonetheless, even expression data, people have shown that there are significant differences with respect to ethnic background and race. And of course, the majority, unfortunately, the majority of the studies are Eurocentric. And it's something that we're working on. So for instance, one of our latest studies is a study in lupus, which is another autoimmune disease. And we specifically were looking at an Asian population and a Caucasian population and looking at differences with respect to clinical parameters, but also with respect to molecular parameters. So yes, my hope is that we will be generating more and more data. In the genetics space, we've done better, especially like I was mentioning that reference data set of 1,000 genomes where they've taken genomes from different ethnic groups from all over the world. But with respect to disease, there's a lot more we need to do. Absolutely. Other questions? Yes. Well, I mean, unfortunately, I don't know how diverse clinical trials are either. So that's something to think about and kind of investigate, because I don't, I mean, even if you look at the latest COVID-19 vaccine trials, they have not looked at all populations or even all, they were all comer volunteers who were parts of those trials, right? So for instance, my dad has an autoimmune disease, but there hasn't been a formal trial in people with autoimmune diseases. So yes, absolutely. Another question: when you look, for example, at the Caucasian population, do you partner with universities and research institutions in those countries? Yes. So for instance, we have a project in endometriosis, which is an inflammation of the uterine lining. And we have samples coming from all over the world. So we have a core set of samples coming from UCSF, but we also have samples coming from Australia and Europe. Again, we're missing a lot. We don't have anything coming from Asia or Africa, but yes, absolutely. We try our best to gather as diverse of a population as we can. Excellent. Other questions? Okay, well, I would like to spend a few more minutes talking about the application of this approach to identify new therapeutics for COVID-19. And this is one of our latest projects in the lab, and it's available online on bioRxiv. If you're interested in the paper, you can look it up. It's led by 3 people in my lab. So it's truly a collaborative effort, and a lot of people in the group came together, but these 3 individuals have contributed the most. So Brian Lee, Guy Androletti, and Tomiko Oskotsky. So again, as I said, what do we do? First, we go out and look for data. So we identified 3 relevant data sets for COVID-19. They're all diverse. The first one is epithelial cells infected with the virus, and we identified 120 genes, 100 upregulated, 20 downregulated as part of the signature. We looked at organoids from gut tissue, and were able to get about 129 genes out of that. And then we had a small patient cohort. So we started the study at the beginning of the pandemic. So this was one of the earliest data sets that was generated. And we got a lot more genes out of that. Then we looked at the biology across these signatures, and we saw that there were genes that were upregulated and pathways, including interferon response, oxidative phosphorylation, and then a number of ones that were downregulated, including adipogenesis and mTORC1 signaling. But this heat map here shows the pathways, the biological pathways that were involved across all of these signatures. Then we wanted to look at the drug profiles, and we followed exactly the same pipeline that I just described. And we saw that across the 3 signatures, which are shown here, first we saw some drugs in common, which was great. And secondly, we saw some drugs that other people have been identifying as well. So haloperidol and valproic acid have been identified by another group, and colchicine at that time was in a clinical trial. So this made us very hopeful. We explored a little bit further, and this was the summary of our findings. These are top 25 drugs that reverse at least 2 of those signatures. And you can see that there are 4 compounds that reverse all 3 of them. This is a network visualization of the same data. These are in pink, the drug hits, and in blue and yellow are the targets. And the yellow targets are targets that are shared across several drugs. So in terms of the drugs that reverse all 3 signatures, these are those top 4. So Bacampicillin is used to treat bacterial infection. Clofazimine is an anti-mycobacterial used to treat leprosy. Haloperidol is a psychiatric medication that's used for schizophrenia. But interestingly, we know that it combines these 2 proteins, sigma-1 and sigma-2 receptors. And it has been found to decrease viral growth in SARS-CoV-2 infected cells by a previous study. Valproic acid is another one, another drug. It's used to treat epilepsy. It's implicated in inhibition of HDAC2, but others did not observe a decrease of viral growth after treating with this drug. So we launched several validation studies based on this effort. This was a collaboration with Dr. Melanie Ott at UCSF and the Gladstone. And these are all the drugs that we looked at. And so in addition to the 4 that we were interested in, we also looked at remdesivir as a positive control. So what you can see right here is a dose-dependent reduction in viral load for treatment with haloperidol, which was very interesting. And you can see the same thing looking at the graphs here. So these 2 are controls, and this is looking at remdesivir, which get rid of all the green dots, and haloperidol reduces them significantly. We launched the second validation study. This was with Adolfo Garcia Sastra's group at Mount Sinai. And here we're looking at reduction of this viral infection load shown in red. So basically you want to see the red curves go down, while you want the black line, which is cell viability, how cells survive, to stay flat. And you can see that actually a number of compounds seem to be having an antiviral effect, including Bacampicillin, cyclosporin, clofazimine, and here is a bunch more that we ended up testing. Tretinoin, of course, remdesivir is a positive control. So this gave us a lot of confidence. Of course, there's additional testing that needs to be done, but here we developed a computational drug repurposing pipeline. I've shown you the initial applications of this pipeline to Crohn's disease, but our most recent work is relating to COVID-19, where we identified 25 common hits and 4 consensus hits, and then validated them in 2 sets of experiments, where we've shown that 11 out of 16, we weren't able to test all 25 because of compound availability, but we did see that 11 out of 16 common hits were showing antiviral activity, as well as 3 out of 4 consensus drugs. And if you think about it, this sort of success rate, even though there's more testing that needs to get done, is quite incredible. Because as I was saying, 90% of drugs fail in early development. And here we have about, 75 or so percent of at least showing some efficacy. We're exploring combination therapy and more advanced machine learning approaches. And as I gave you an example with Anushrie's project, we are also leveraging electronic medical records data to try and validate some of these findings. So in terms of electronic medical records data, it's very complementary to the molecular data sets that I've mentioned. At UCSF, we have data from 2012 until today, and it includes diagnosis codes, lab test results. So pretty much if you go to the doctor, your whole medical record can be de-identified and used for research purposes. And Atul Butte, who is my mentor, has been leading this effort to aggregate this data across the state of California, which this says 12 million. It's actually over 20 million patients. So it's a huge, huge data resource. This is the data at UCSF, just showing the demographics. But nonetheless, we have other types of data like diagnoses and medications and labs as well. And specifically for COVID-19, there has been this incredible effort by a group called Cerner to aggregate these clinical data. So this is a data set that we have access to and we've been working with that has over 120,000 COVID-19 patients, as well as their full medical history. This is across 62 institutions in the US, roughly split equally between males and females, and all the same data that I've been mentioning before. Clinical data is available on these individuals. So the hope is by combining the molecular piece, which I spent a lot of time today talking about molecular data, together with the clinical measurements, then we can get a little bit closer to precision medicine. And with that, I'd like to thank my team at UCSF. All the COVID-19 work was led by Tamiko, Brian, and Gaia, with help from others who are underlined here. The collaborators on the IBD work are in this here in these two columns, and then these are our wet lab collaborators for validating the COVID-19 hits. I'd like to thank my funding, of course, because none of this would be possible without the support from NIH and other institutions that fund our work. This is us actually a year ago, almost to the date, where we all went out to lunch, I think our last time, all in one elevator. And here we are now on Zoom, just like everybody else. So with that, I'd like to thank you for your attention, and I would love to take any additional questions. Okay, so there's a question from Ryan. If I enjoy both math, chemistry, and bio, would this be yes? Yes, 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 absolutely. I find bioinformatics incredibly exciting. I think it's a way to apply computational skills to problems that are incredibly relevant and can impact the lives of patients. And I think it's very powerful and also very exciting because you're really, with research, you're really working on the edge of what's actually known. So it's a lot of fun to be in that spot. Yes, so question if I am hiring and taking volunteers virtually. Yes, so every year I have a number of high school volunteers in the lab. We are probably almost full for the summer, but do reach out to me if you're interested. And the other thing I do is I run this program called AI for All at UCSF. So hold on, sorry. I will paste the link in here. It's a 3-week summer program. And the goal is to introduce high school students to applications of artificial intelligence in biomedicine. So check it out if you're interested. The applications go live on March 1st, so there's a little bit of time. Excellent. Other questions?
Okay, so we actually have some discussion questions we would like to discuss with you.
Okay.
Okay, so first question is, how can high school or post-secondary students get involved with AI? So like, are there any other opportunities that you would suggest to high schoolers or post-secondary students?
So AI for All is one that I know a lot about. The program started at Stanford. I was involved with it at Stanford at the beginning, but now many universities around the country have it as well. There's a question of whether the application is open to Canadians. That's a great question. I don't know the answer to that, even though I should. Send me an email. There actually might be a school in Canada that has the program, but I'm not sure. I can find out. Yeah, so that's one way. So look out for these programs or internship opportunities. Cold emailing labs sometimes works. It's hard. PIs are really, really busy. Oh, the program is free. Our program is free at UCSF. There was a question about that. Oh, I will paste my email. And our lab is, this is our lab website. Sorry, there are probably, internship opportunities and intern programs in various universities. Not all of them are free. I'm very passionate about keeping our program free. But there are ways to volunteer in research labs if that's something that you're interested in.
Oh, okay, that's great to know that other students can, sorry, can participate and have some interaction with this area. So what would be like advice that you would give yourself if you were looking back on your high school and post-secondary career?
That's a good question. Well, first I would say don't stress, everything will be okay. You will find yourself and find something that you're excited about and it'll be great. Of course, now it's a difficult time with COVID and everybody being home all the time, which I can't imagine what you guys are going through. It must be really rough. But hang in there, find something that you're really passionate about, whether it's science or finance or history, whatever it might be, or art, and ask what the big problems in that field are, and then don't be afraid to connect with experts. I mean, I still do this. I'm not a clinician, but every single project that we work on, we have a clinical collaborator to understand those questions better. So find people, experts in whatever field interests you, and ask them to be your mentors, learn from them, take advantage of that. People like to share their experiences and help others. So I feel like those are all good things to keep in mind. There's a question about whether the program is open to university students. No, the AI for All programs are high school programs, as far as I know, but I'm sure there are other university level programs as well.
Yeah, so I think the students can do some research or maybe like searching on the web to find some opportunities.
Absolutely.
Yeah, okay, so also regarding like the current pandemic, has it presented any obstacles to your current work and how does it affect it?
Well, my work is all computational. So you can see I'm in my bedroom office right now. So I'm working from home. And we have been working remotely for the past year. So for us, I feel
Yeah.
like we're incredibly lucky. We have not been affected too much. Of course, I worry about, my team and my students and burnout and the fact that, folks are just mostly at home. So those are all concerns, but overall, the group has been doing incredibly well.
Yeah, that's great to hear. The pandemic has affected quite a bit of different people and especially students. I think they're affected by not being able to have like live classes, and actually it's pretty hard. Yeah.
Yeah,
So
I can't imagine.
yeah, it is. So I mean, so since the pandemic doesn't like affect it too much, so did you ever have like a great setback or like a large setback on your work, and how did you like respond to this setback?
I wouldn't say a large setback on like work overall, but within individual projects, for sure. For instance, we had a genetic study studying outcomes, pregnancy outcomes, specifically preterm birth. And it was a very large study. It was almost 17,000 people, genetics of about 17,000 people. And we realized, because the data was coming from different sources, that we had a bias and we made a mistake. And we caught it in time before the work was published or presented too widely. And it was difficult. I mean, I had to get up basically in front of a lot of people and explain why this was a difficult problem and why our initial conclusions were not right. But we overcame that. We learned a lot. We realized that maybe certain data you can't use certain data to answer certain questions, and that's okay. And sometimes a negative result is just as important as a positive result. So yeah, I mean, that's an example of one study. But within each study, there are small setbacks. This was a big one because we realized we had a big mistake and we needed to figure out how to correct that and understand where it's coming from. But yeah, absolutely.
Yeah, yeah, I think that totally makes sense. So the next question would be, what do you consider to be the most significant contribution with your work? So how do you think it makes like a difference in healthcare?
Well, I think that the drug repurposing work that I was mentioning today, has made the largest impact probably to date. And I'm hoping again that it's a way to generate hypotheses, to show that you can use data and computational methods to generate these hypotheses and then go on and test them in the lab or clinic or whatever the next steps might be.
Yeah, okay, that totally makes sense. So I think there's a question in the chat that says, any advice for getting into bioinformatics? I don't have too much knowledge in the subject. So grade 12 course recommendations, I think is what he's asking.
Yeah. So I would say beef up on statistics. I know statistics sounds boring, but it's actually quite important. So I would recommend learning a little bit about statistics and only have grade 12 knowledge. Yeah, so statistics would be one area. And then try playing with data. Like you can go online and just download some of these data sets. There's a lot of, if people are inclined to do machine learning or image analysis, there's tons of imaging data out there. There's a lot of expression data. TCGA is a great example. It's a cancer data set that I mentioned. And just play with it, see what that data exploration, where that leads you. And also look for a mentor, look for somebody at a university nearby who might be interested. You can either work with the bioinformatician of the start or work with a biologist to ask what the questions are, and then you can try and do the analysis on your own and kind of identify the methods there. There's also probably Coursera courses on bioinformatics that you could take that are free.
Yeah. Okay. So
listen to some TED Talks. Those are fun.
Yeah. Yes, that's a good recommendation. Okay, so I think he has another question. He says, I guess cold email.
Try. I mean, it's best to go through connections, but it's always worth a try. And don't get discouraged. You got to send a lot of cold emails to get somebody to answer. I don't know, 20, 30. Don't get discouraged if you don't hear back.
Yeah, totally agree. Okay, so yeah, so thank you for everyone's questions. So if you guys have any further questions, maybe email Dr. Sirota.
Yes, absolutely.
Questions. Yeah. So I think
inviting me.
Yeah, and then she'll put her email in the chat, maybe.
Yeah, so. I already put it in the chat and the lab.
Okay, great.
AI for all website.
All right, great. Okay, great. Thank you. So thank you so much for your presentation today, Dr. Sirota. And I hope that you all enjoyed the presentation, and we'll